Alright guys, welcome back to Underground Science, and in this video we'll be talking about the phosphatidyllinositol 4,5-biphosphate, or also called Pip2, and inositol 1,4,5-triphosphate, also called IP3, and the diacylglycerol, also called DAG, the second messenger pathway. Alright, so let's go and get started. So this is pretty much, if you went and watched my G protein coupled receptor pathway, right, the protein kinase A pathway, it's not the same thing as this, but it's still a second messenger pathway that's responsible for conducting mechanisms intracellularly. All right, so something's going to bind as a first messenger outside the cell on a G protein coupled receptor, right? But it's not going to be a G stimulatory protein this time. It's going to be a GQ protein. So it's going to involve a GQ protein, unlike our video that we made on the G stimulatory protein coupled receptor pathway. All right, so it's going to be a GQ protein, not a G stimulatory protein. All right, and there's going to be many other things, but this is just the starting. So over here, what I did is that I drew a regular cell. Okay, so a regular body cell. So let's go and write cell. So a regular cell, and this is the cell membrane. All right. So what I did is that I zoomed in on the phospholipid bilayer of the cell membrane, right? So about on this part right here. All right, so this is a zoomed in version of that. And so I want to take a closer look, because if we zoomed in on this part, that means all of this part right here is going to be the outside of the cell, and all of this part right here is going to be the inside of the cell. So we go and write down the same thing, that over here we're going to see that we're talking about the inside of the cell, and over here we're going to be talking about the outside of the cell. All right, just the same, this is proportional to this, except it's much bigger, all right, down here. So let's go and get started. So the first thing we have to talk about is our G protein coupled receptor again. All right, so let's say we have a G protein coupled receptor here. Let's go and draw it again like we did in the previous video. So a G protein coupled receptor, let's go and draw a receptor, and let's go and finish this, right? So let's go and draw that, and then we're going to have our ending here. So this is our G protein coupled receptor, but this time we're going to have not a G stimulatory protein in this specific pathway, right? We're going to have a GQ protein. So we're going to have a GQ protein here. So let me go and write this down more neatly. So we're going to have a GQ protein, a GQ, there you go. And this is going to be part of the alpha subunit. All right, so it's going to be a GQ alpha subunit. That means that we're going to have a beta subunit and a gamma subunit here. So what you'll see is that we actually have a three subunit protein bound on the G protein coupler. So we're going to have a GQ alpha subunit, we're going to have a GQ beta subunit, and we're going to have a GQ gamma subunit. All right, so we're going to have a GQ gamma subunit. All right, so now that's over with, we're going to see that the first step pretty much is going to be the same as it was in the G stimulatory protein coupled receptor video. All right, so what's going to happen again is that, let's say we're going to get a peptide hormone or some type of ligand that can't just cross the cell membrane, and it has to bind to our G protein coupled receptor. So let's go and draw some type of hormone, a peptide hormone in particular, right? Or if we have to generalize it, we'll just say it's of some type of ligand, and this will count as our first messenger ligand because it's outside the cell. All right, so this is our first messenger ligand. So first messenger ligand. Now what's going to happen is that once it binds, again in the previous video, in our other G stimulatory protein coupled receptor video, we're going to have GDP bound onto our GQ alpha subunit. Okay, but the second we have something bind, a ligand bind to our G protein coupled receptor, what's going to happen, guys, is that our GDP is going to combine with an inorganic phosphate and not be guanine diphosphate anymore. It's going to be GTP. All right, so it's going to have one extra phosphate. That extra phosphate addition is going to give this GQ alpha protein some energy to leave this party right here with the beta and gamma subunits, and it's going to leave that party and travel to a nearby enzyme. Okay, so it's going to travel to a nearby enzyme. So let's go and draw that. So what we're going to see is that it's going to dissociate, right? It's going to leave the party. So like, I'm out of here, right? I've had enough. And let's go and finish drawing this, right? So we're going to dissociate, and we'll see that we actually have a nearby enzyme here in the cell membrane, all right? That's going to be kind of like a phospholipase, right? It actually is a phospholipase. So it's going to be called a phospholipase C enzyme in particular, all right? So we're going to have a phospholipase C enzyme. So let's go and write that out. Sorry about this, Mark. All right, so it's going to be our phospholipase C. All right, so phospholipase C enzyme. Now, what is this phospholipase C enzyme going to do? Well, where's our GQ protein, GQ alpha subunit, right? So remember, it has our GTP bind to it, and it moves along the membrane and binds to our phospholipase C. All right, so our GQ protein, let's go and write it here. Our GQ protein, right, will come here and bind to the phospholipase C enzyme. We can't forget our GTP bound to it. All right, once it binds to the phospholipase C enzyme, the GTP will get hydrolyzed into GDP and an inorganic phosphate. And remember, initially, when our GQ alpha protein was bound to here, we had GDP on it, and GDP kept the GQ alpha protein inactivated. So once the GDP is formed from the hydrolyzation reaction of GTP, GDP is going to go and inactivate our GQ alpha protein. All right, so once our GQ alpha protein, however, binded though in phospholipase C will get activated first, and then our GQ protein will get inactivated by our GDP. All right, so once our phospholipase C protein, or our enzyme, is activated, what is this going to do? Well, it turns out we have another compound or another protein bound in our phospholipid bilayer near our G protein coupled receptor and near our phospholipase C. What is that molecule called? Well, it's kind of a mouthful, all right? So let's go and draw it out first. So let's say this molecule, or this compound, this whole protein complex, right, is right here. Just go and draw something like that. And let's go and shade this in. So this whole protein complex here is going to be called PIP2, all right? So it's our Pip2 protein. Now the full form of this, if you guys want to know, is kind of a mouthful. So it's going to be called phosphatidylinosytol 4,5-biphosphate, all right? So phosphatidylinosytol 4,5-biphosphate. Really hope I spelled that right. All right, guys, so again, this enzyme right here, this protein is on the phospholipid bilayer, is going to be called Pip2, or phosphatidylinosytol 4,5-biphosphate. All right, so it has a phosphate group on the 4th and the 5th carbon. All right, so that's why it's a biphosphate. All right, so that's that. So when GQ alpha, right, when our GQ alpha protein binds to this phospholipase C will actually cleave phosphatidylinosytol 4,5-biphosphate into two really important proteins. What are those two proteins? One protein is going to be called IP3, and one protein is going to be called DAG. All right, so IP3, guys, stands for inositol 1,4,5-triphosphate. So inositol let's write it different. Inositol 1,4,5-triphosphate, and the reason why it's triphosphate is because we have a phosphate group in 3 places, right? We have a phosphate group on the 1st carbon of inositol, 4th carbon of inositol, and the 5th carbon of inositol. All right, so it's inositol 1,4,5-triphosphate. And our other molecule, DAG, stands for, let's go and split this down right here, stands for diacylglycerol, right? In particular, 1,2-diacylglycerol. So 1,2-diacylglycerol, you can call it also. All right, diacylglycerol. All right, so you can call it 1,2-diacylglycerol, or I just call it 1,2-diacylglycerol. All right, so hopefully I'm not pronouncing it wrong. So 1,2-diacylglycerol pretty much just means, like, remember from fatty acids, when we talk about digestion of fatty acids and all that, we have triglycerides, or also called triacylglycerols. Just like that, we have a diacylglycerol here. So that means that diacylglycerol has 2 fatty acid chains. So 2 fatty acid chains. All right, so that's what happens. So we have to say that. So what's catalyzing this reaction right here? Well, the splitting of phosphatidylinositol 4,5-biphosphate into inositol 1,4,5-triphosphate and diacylglycerol, right? 1,2-diacylglycerol. This enzyme that does that is our phospholipase C enzyme. All right, so phospholipase C. All right, so now what happens, let's go back up here. So it turns out that 1,2-diacylglycerol is actually hydrophobic, and it actually likes to stay bound to the phospholipid membrane. All right, so the phospholipid bilayer membrane. So let's go and draw our diacylglycerol right here. All right, so let's go and say that our diacylglycerol, right, is right here. All right, so I don't know why I drew it like that, but let's just say it looks like this, and this is our DAG that's bound to the phospholipid bilayer. Remember, guys, this is the inside of the cell, and this is the outside of the cell, right? So it's just to zoom in over here. So we can write it here. This is the inside and the outside of the cell. So we have another organelle on the inside of our cells, right? That organelle is going to be our smooth endoplasmic reticulum. All right, so let's go and draw our endoplasmic reticulum, or at least attempt to, right? So let's say this is our endoplasmic reticulum, right? And in our endoplasmic reticulum, we're going to have calcium. So let's go and draw one endoplasmic reticulum, especially a little bigger, and we're going to have some calcium, all right, in our endoplasmic reticulum. And if this is our muscle cells, we're not going to have an endoplasmic reticulum. I can't talk today. We're going to have a sarcoplasmic reticulum. All right, so this is going to be our endoplasmic reticulum that's going to have calcium stores in it. All right, and then let's go and label this. So this is our endoplasmic reticulum, and we should write that there's an exception where in our muscle cells, so let's just go and write it here, right? In our muscle cells, we're going to have not an endoplasmic reticulum, but we're going to have a sarcoplasmic reticulum. Sarcoplasmic reticulum. All right, that's going to be responsible for storing calcium, which is important for contraction of muscles, which we'll go over in a future physiology playlist. All right, so what will this endoplasmic reticulum do? Well, you see this IP3 that was produced? IP3 turns out to be hydrophilic, so it dissolves. It doesn't dissolve, but it likes to be in the cytoplasm of the cell. All right, so it's floating out around right here, and it comes over here and kind of binds to a channel that's present on this endoplasmic reticulum. All right, so let's say the channel is right here. All right, so the channel is right here. I mean, it has a receptor and a channel, right? You can think of it like that. And what's going to happen here is that once IP3 comes right here, actually, you know what? This is getting too crowded. Let's go ahead and erase some of these right here, some of this, and write it neater. So again, this is our sarcoplasmic reticulum, and this is our PIP2, right? And we don't need the full form anymore because we already discussed it, phosphatidylinositol 4,5-biphosphate, all right? So this is our receptor for IP3, and this is our calcium channel. So what's going to happen here is look at this. So we have DAG here on the membrane. We'll see what it does in a second. What about IP3? What does it do? Well, once IP3 is created, right, because of our phospholipase C enzyme, right, it breaks down. Oh crap! It breaks down right here. Should write it down. It breaks down our PIP2, our phosphatidylinositol 4,5-biphosphate, into IP3 and DAG. IP3 will come. Watch this, right? IP3 will come bind to the receptor on the endoplasmic reticulum. This binding will cause will cause our calcium to start pouring out into the cytosol, into the cytoplasm of our cell. So this is still the inside of the cell, right? So we're going to have calcium starting to pour out of our endoplasmic reticulum, right? Sarcoplasmic reticulum if it's our muscle cell. Now, what good is this? So calcium, right? The calcium that starts pouring out will go here. It will combine with our 1,2-diacylglycerol, right? It'll combine with our 1,2-diacylglycerol. So let's just write it. Calcium has combined, and it's going to go later. It's going to go down the cell membrane and find an enzyme called protein kinase C. All right, so let's go and show that. So calcium and 1,2-diacylglycerol will go down and find our protein kinase C, which will be right here, right? Our protein kinase. Remember in our G-coupled protein receptor video, our G-stimulatory protein in particular, we talked about at the end of protein kinase A being activated. Well, here in our Pip2 pathway, it's going to be a protein kinase C that's going to be activated with the help of our calcium bound to our 1,2-diacylglycerol. So this whole complex as a whole with calcium and 1,2-diacylglycerol will come here, activate the crap out of protein kinase C, and now our protein kinase C will be our intracellular protein that will phosphorylate proteins in order to carry out whatever the cell needs to carry out. All right, so this guy will go ahead and phosphorylate stuff, okay? And we'll see in later videos, like angiotensin 2 will come to our zona glomerulosa, right, of our adrenal cortex, which we'll go over in a future video. And it's actually going to follow this pathway in order to help make a molecule or a cholesterol derivative called aldosterone, right? So it's going to be made from cholesterol, and we'll see how it works in this Pip2 pathway. All right, so hopefully this video made sense to you guys. Hopefully this video wasn't too boring or confusing. Hopefully you guys got the basic idea of this Pip2, our phosphatidylinositol 4,5-biphosphate pathway. That's the full form. Our inositol triphosphate pathway, or inositol 1,4,5-triphosphate pathway, and our 1,2-diacylglycerol pathway. And this all worked through a second messenger pathway via intracellular proteins, all right? And that main protein was protein kinase C. That was going to carry out many functions inside the cell by phosphorylating, all right? Because it's a kinase protein. And it's going to, remember, calcium is important here because we're talking about the endoplasmic reticulum for regular cells, but for muscle cells, we're talking about the sarcoplasmic reticulum. All right, and so hopefully this video made sense to you guys. This is the end of the video. Make sure to tune in into my future videos where I'll talk about more cool topics about biochem, physics, and chem. Make sure to hit that notification bell, like, and subscribe, and see you guys later, and stay safe.